Okay, good afternoon, viewers at home. Sorry for that delay in the coming up. We are continuing our educational series on genetics, and now, just like what we did the other time, we are continuing on non-Mendelian law, Mendelian principles. Okay, this is exactly what is called deviation from the Mendelian principles. The last time we listed them as incomplete dominance. That's number one. That is a kind of deviation. And that day I made mention of um, a plant that is a snapdragon plant, where we have when you cross uh, a red plant, you cross it with a white plant. And don't forget, I said all, according to Mendel, all red, that is after first filial generation, all will be red. So, and to get our F2, we are going to cross the second filial, first filial generation with that. And what we observe is that we have, sorry, so we now have this to be red, this to be pink, and we said this one to be white. So the question now is, where is this one coming from? Since the parental, this one is red, parent, and this one is white. So example of this is a non-Mendelian crossing. So that is the first case. non-Mendelian crossing. Okay, don't forget that the second one, is a co-dominance. So the first one, which is on this, you need to know that it is incomplete. So you have an intermediate. You have production of an intermediate, which is combination of red and white, which is going to give us a green. I mean, sorry, which is going to give us pink, just like what we have said. So, and I said this one happens in a plant which is called snap dragon. Okay. So the second case is co-dominance, the law of co-dominance. Okay. So this is the example of this is what we see in this is a situation where both allele, you need to know, both allele are equally present. Both are equally present, and apart from that, um, the resultant combination of the two, make it very very feasible. Make it of the two, make it feasible physically. It's going to be present physically. Okay, so that is why now. Okay, so this is the first case. So the second case is co-dominance. The two allele, they are present equally. They are present and represented equally. That is co-dominance. So, and that one is what we have. That one is exactly what we have in the case of ABO blood grouping. I told us in the ABO blood grouping. Okay. Then apart from that, we have a situation in N. We have a situation that is still co-dominance. We have it in N. So some N, while some appear to be black and some speckled. So those ones that appear phenotypically, you have to know, phenotypically, those ones that appear to be white, they have phenotypically, those ones that have, they are W, that is white, that is capital letter. Those ones that are black, they are found to be this, and those ones that are speckled, they are found to be this. So if you now look at these three, you already have three: white, black, speckled. So this is a kind of deviation from the Mendelian principle that says one trait or one allele will serve as dominant and the other one will serve as a recessive. So in this case now you have three types. You have W, which is this, and you have those ones that are black and those ones that are this. So under the same condition, now if you now want to cross white, you want to cross it with black. That is, this one is W, we are crossing it with black, so that at the end of the day what you are going to see is BW, which is going to be heterozygote BW. This one is a kind of combination, just like I said, it's a kind of combination of white and black, and you just discover that all of them will appear to be speckled. All of them will appear to be speckled. Instead, don't forget the law of Mendelian law. This was supposed to be heterozygotes, heterozygotes, and once they are heterozygotes, now we expect that for them to appear black, but they appear speckled. So when you now cross this for the F2, when you cross it together again, so that you are now going to have BB and BW. This is still under incomplete dominance, so that these two will appear speckled. This one is going to still retain black, and this one will still be white. This is still another case of a law of incomplete dominance. Incomplete dominance. Okay, so we were supposed to look at a case of co-dominance. A case of we find out that case of co-dominance happen in cow, where you have a white cow is being represented as this. This one is white, and some in that have something like this is called Rowan. So that by the time you now cross this together, let's say you use your Punnett square, get your Punnett square, this place is going to be female and male gametes. So this is going to be CR, CR. So that at the end of the day, when you now cross, you are going to have CWCR, CWCR. Then you also cross CWCR, CWCR. So instead of it to appear to be like what you normally find, this one is found in coat color. So we have, this is supposed to be red. This is red. So anyone that has CWCR is going to be Rowan. So instead of you to have either white or red, the third color is now Rowan, so that all the organisms in the first filial generation, all of them will appear Rowan. So that when you now cross them together, when you cross this one, F2, so that means you are going to cross it so when you cross them together, that is CWCR cross CW dot CR, so that when you now do a Punnett square crossing, CWCR, okay, CWCR. So let's look at, we are going to have CWCR. Okay, this is going to be CWCR. This is going to be CRCR. So, and interpreting this, I mean that At the end of this crossing, this is going to be white, this is going to be Rowan, this is going to be Rowan, this is going to be red color. So it is still going to be 1, 2, 1. That is co-dominance, because the law of co-dominance, because both these allele, they are equally expressed. That is what is called co-dominance. Okay. Now, the other one that we will also look at, that we examine that time, was the issue of multiple allele. And I actually told us about example of ABO block grouping. Okay. ABO block grouping. So you know how ABO block grouping can be expressed. So the earlier video we had, I was unable to actually demonstrate how ABO block grouping is actually is being inherited. ABO block grouping. Okay, don't forget that I told you that that is multiple allele. Is either you have, so if you have something like this, this is red blood cell, another red blood cell, another red blood cell, another red blood cell. Okay, so inside that red blood cell, you now have, if this one is A, group A, this is containing allele B, this is containing both A and B, this is containing none. So what that means is that if antigen A, let's say this is antigen A and this one is antigen B, this is, let's say this is A, sorry, if this is B, if this one is A. So what it means is that on A, we have this antigen. On B, we have this antigen. And on AB, we have both. So on O, none of this is possible. Now, don't forget now, inside the serum, we now have what is called antibody. That is what is going to determine who is going to collect blood during the process of transfusion without any problem. Don't forget that if B comes here, it's going to be a problem. Look at this shape. If B comes here, there's going to be a problem. The only thing is that A will get from A. If A come here, there's going to, so for A, for anti-A now, that means it's going to have anti-serum. That means B is going to contain anti-serum B. This one will have A. This one is going to have both. And this one is going to have none. So what it means is that since this one doesn't have any, so if this one is to act as a donor to any, so this one we have no problem with any. That's why this one is regarded as a universal donor. And if it is this one, because this one has anti-A on it. So if A is coming in, it has something to receive it. If B is coming in, it has something to receive it. If AB is coming in, it has something to receive it. And if this one is actually coming, if this one is actually coming in, that is when there's going to be any no problem. So this one can receive four. That's why they call this one universal recipient, whereas this one is a universal donor. So pattern of inheritance, the pattern of inheritance of ABO blood grouping actually follow that one which is the law, which is a deviation from the Mendel principle. Mendel principle. Don't forget what I've given you cited example now. I gave you examples of incomplete dominance, whereby I mentioned snapdragon with the botanical name, which is Antirrhinium majus. Snapdragon is Antirrhinium majus. So they discovered that that plant do not obey Mendel's law because it has some plants with white flower and some are red. So during the process of cross fertilization, so they now discover that the first filial generation, that was RR, under normal circumstances this was supposed to appear to be red, but instead of it to be red, it appeared to be pink. So when you now cross them together again, so you now have 1 2 1, you now discover that red was in ratio 1, white was in ratio 1, but pink, which is the third color, was also in ratio 2. That was in incomplete dominance. Then codominance, that is where you have both allele, they are equally present and equally expressed. So, and I cited example of ABO blood grouping, and I also cited example of what we have in N, where we have white, we have speckled, so still under codominance. Also in the law of ABO blood grouping, which is a multiple allele, you also have something that also look like that. That is when you have, so that is where you have any of the three allele is possible. That is either A or B, and if it is containing the two, that is AB. If it has one, it could either be A or B, or if it has none, it's going to be O. So, and I've actually told you how some blood group are classified as universal recipients, while some are regarded as universal donor. So the next thing is polygenic genes. That is number 4. Polygenic genes or polygenic inheritance. Just like from the name poly, that's telling you that you have one gene, you have one trait, you have one gene going for different traits, that is more than one trait. Under normal circumstance, it was supposed to be one gene, one trait, but in polygenic genes we have more than one trait that are expressed by one gene. That is why they call it polygenic inheritance. That is one gene to one gene to several traits. Okay, so an example of that is found in wheat. Polygenicity or polygenic inheritance is found in wheat. So, and what happened in yeast is that they propose that at least two genes, which A or B, so this A or B will be dominant, not one now. This was dominant, and that means the recessive is going to be A or B. That is recessive. Okay, so that by the time they now use a Punnett square to cross them. So example of polygenic gene, human height is also an example. So that when you now use A, this, so you now have A, B, you are crossing it with another A, B, so that this one will now be anywhere you see A or B, this capital this or this is going for red, and anywhere you see any of this one is actually going for medium. So, and anywhere you see A or capital A or this, or you have it like this is going to be dark. This is always dark in color. So, and if you look at if you use Punnett square to determine what is going to be the product of crossing this with this, you will know. Let's see. Let us see. So this is going to be A, B, and A capital B and A, B. When you also cross it with another A, B, another A, B, another A, B, then another A, B. So that you have A, B, B, you have A, B, B, you have A, B, B, you have A, B, B. Then this one A, B, B, A, B, B. Then this one A, B, B. Then this one too A, B, B. Then A, B, B. A, B, B. You have A, B, B. A, B, B. So this is going to be A, B, B. This is going to be A, B, B. A, B, B. And you have A, B, B. So let's now interpret. I've mentioned three. Okay, I've mentioned two examples of where you can have polygenic genes in human height. You also have it in the kernel color. So this is going to appear red. This is going to appear dark. This is going to be dark. Okay, this is going to be medium. Medium. This is going to be dark. This is going to be medium. This is going to be medium. This is going to be medium. Okay, this is going to be dark. This is going to be medium. 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 So when you now count medium, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11. When you count dark, 1, 2, 3, 4. 1, 2, 3, 4. That's 4. And if you count red, it's going to be 1. So this is medium. This is going to be dark, and this is going to be. Now discover that instead of it to be 9, 3, 3, 1, it does not obey that, and that is an example of a polygenic example of where you have a polygenic inheritance. Polygenic inheritance. So don't forget that I told you that wherever you the dominant traits in any of these plants and some other, anywhere you see polygenic gene, so it's going to be. So if you are not given any genetical problem, maybe in your test or any exam, if you are given any genetical problem that is polygenetic or polygenic inheritance, if you get this kind of ratio 11:4:1, you should know that that is talking about polygenic inheritance. That's polygenic inheritance. This one is recessive. So, and that's why the presence of it, wherever you see the presence of it, does not really get expressed or get dominant in that case. Okay. So I think you should know that as well. Now, number 5 now. Number 5, which is a case of okay. Number 5, which is a case of sex linked. Let's look at example of sex linked genes. What really happens in case of genes that are linked to certain sex? I told you that there are some genes that are only unique with respect to X, and that is that. For example, now in sex determination, in sex linked. So okay, number 5, which is a sex linked inheritance, it's also a kind of a deviation from Mendelian principle. And example of that is that there are some that are called X linked recessive disorder. That is, you found some diseases or disorders in certain sexes, like baldness, is associated with chromosome Y, and that of hemophilia, this one is linked with X chromosome. That is linked with X chromosome. So, for example, if you have, let's say you have, if capital H is normal and small h is abnormal, that is hemophilia abnormal. So if you now have that means if you have anyone with this, anyone with this, that means the person is affected. The person is affected. So when you have a male, let's say you have a male which is normal, male normal, male normal, do not carry any gene at all. So we are going to see this later on. So if this person is a normal male marrying somebody who is affected, let us see capital which is normal and age which means is affected. So when you cross, do the crossing by the use of your Punnett square. So this is going to be H. You are crossing it with Y. This one, you are crossing it with this. So when you cross this with this, you cross it with this, cross this with this. So cross this with this. So what this is going to tell us is that this one will be normal female, because the person is a XX, chromosome XX. This one is a affected male. Sorry, affected female. This is affected female. This one is normal male. This one is affected. No. This one is abnormal. This is abnormal. Abnormal male. So if you want to trace, so this person will be abnormal male. If you want to trace the origin of this age, you have to trace it back to the female. So that is why the gene of hemophilialism is actually being determined by its sex linked, the sex linked inheritance. So such is what is very, very possible. So that is number 5. Number 6 now. Number 6 is going to be mitochondrial inheritance. Mitochondrial inheritance. So there are certain that is mitochondrial inheritance. So there are certain genes. Don't forget that I said mitochondria is an organelle that contains its own DNA. It contains its own DNA, although and which has the ability to replicate semi-autonomously. So it might not actually go in line with everything that happens in the cytoplasmic DNA. So it has its own DNA, making it to certain level independent, to certain level. It's just like the supply of electricity. You see, we have the main dam is located Kanji Dam. That is where electricity is being generated. And you now see some substations, some dams, which are feeder dams. So it's just like mitochondria is also a kind of a feeder organ, the cytoplasmic DNA. So because this DNA is DNA, and DNA is known to carry on certain genes on its own. So they now discovered that there are some genes that are also located in mitochondria, and the pattern of inheritance of such genes will not follow the normal Mendelian law of dominance and recessive. So example of that is a disease condition that is called Leber's hereditary optic neuropathy. Optic neuropathy. Okay, with the short form LHON. So this kind of a disease condition is located, the genes that is responsible, that is malfunctioning, is actually linked to the genes in the DNA. So if there is any problem in the mitochondrial DNA, this is the condition that it is going to present itself. So irrespective of the sex, the person could be, or it could be female, but what we know is that this kind of inheritance will not actually favor, or will not be in compliance with what is obtainable in normal Mendelian principle of inheritance that has to do with hemophilianism, or that has to do with dominance and recessive. Okay. Condition 7 now. Condition 7, and that is epistasis. Condition 7 is epistasis. Okay, what do we mean if we say epistasis? This is a condition that occurs when one gene is masked. One gene is masked or modified by another gene, which may be on the same chromosome or in another chromosome. That is called epistasis. Epistasis. So an example of that is what we see in mouse. So some mouse can either be, their skin color can be black or F are represented by capital B, and those ones that are brown are represented by small b. This is located on a different chromosome, but this one is known to have effect on a second gene that also control where this color pigmentation will be deposited into. So, and that gene can either be C, capital letter C, or small letter c. So that is why those ones that now have, sorry, this one is small letter, those ones that now have small c, this one will appear to be colorless. Anyone that has B is going to be black. As BB is going to be brown. Anyone that has C, small letter c, will be colorless. So this is a condition referred and called by, that is caused by if one gene in another chromosome is controlling another gene from another chromosome. So such condition is known and called as epistasis. known and called epistasis. Okay, so that is that. Now number eight now, this is epistasis is number 7. Now number 8, that is environmental condition. They discover that the environment has it has a critical role to play. That's number 8. It has critical role to play in determining the genetic makeup or the genetic composition of an organism. Environmental. So for example, the interaction, especially in cats, so they have in cats production of Siamese cats is influenced by environmental factor. I mean temperature is influenced by um temperature. So the gene for, if the temperature is normal, the four color of the four colors they are here, so which can be tail, paw, or here. So if the temperature is normal, so you just discover that their four color is going to be, the gene for four color is going to be dark. It's going to be dark. But if the temperature is warmer, we now discover that instead of the four color to be dark, it will now be Siamese. It will be Siamese. And there, this Siamese color is said to be in a state of inactiveness. It's said to be in a state of inactiveness. Not only that, in cats, we also have it the in human as well. Just discover that if you are from Africa, you have a different color entirely. You have a kind of a genetic color. And normally, if the environment is just too harsh, an organism is bound to adapt to whatever environment it found itself. So that is an environmental condition. Number 9 now, that is anticipation. Number 9 now is a condition that is called anticipation, and that happens in Huntington disease. Okay, am I correct? I think Huntington disease. So Huntington disease is wherever you have repeat in the sequence. If you have repeats of this sequence, CAG repeats, so such an individual that is born with CAG repeats is prone to have a condition that is called Huntington disease. Huntington disease. So it is an anticipation. It might come or it might not come, but it is known genetically that any organism or human that has CAG repeats in their sequences, in their chromosome, they are prone to this kind of condition. And that is a condition where we have anticipation, which is a genetic disorder. A genetic disorder. So if somebody now has this, you will be able to predict the next 2 3 generations, that the next generation of somebody like this with the CAG repeat is going to be suffering from a condition that is Huntington disease. Okay. Now the last one, which is number 10 now. I won't dwell too much on this. That is where you have genetic linkage. I explained it the other time. Genetic linkage or recombination. Genetic linkage or recombination or genetic recombination. That's where you have during meiotic division, when 2 chromosomes come together, join together in the normal process of normal meiotic division, you just discover that there is exchange of genetic makeup, of genetic information between 2 chromosomes, and that is a condition that the inheritance of this condition deviates from the normal Mendelian principles. So don't forget that what we have said so far, we have listed 10 different conditions that will likely arise, and the inheritance pattern might not actually follow Mendelian principle of inheritance, which has to do with one gene uh, is having an allele, and one of these is going to act as a dominant gene, and the allele is going to be recessive. So in this case, we made mention of incomplete dominance, where we mentioned snapdragon. We talked about a co-dominance, that is the presence of the 2 allele uh, is actually very very important, that each of them are equally present and equally represented. So that is a co-dominance. And that of multiple allele, we made mention of ABO block grouping, just like we have inheritance according to depending on whether depending on the blood group of either the recipient or the donor. So the inheritance pattern does not in any way, and it's not going to follow the pattern of Mendelian law. So we also mentioned number 5, we also mentioned polygenic genes, whereby you have one gene controlling poly, that is different types of traits. So, and we mentioned that, I cited example the other time of human skin color. So it is also still very very possible. Then number 6 is a case of mitochondrial inheritance, whereby a certain condition, which is called Leber's hereditary optic neuropathy. This is a condition that can be traced back to the genes of or the DNA of mitochondria. So that is an example. Then again, we also mentioned that of epistasis, which is number 7, which I said that is when you have the present one in one chromosome is controlling another gene in another chromosome. That is a condition of epistasis. Then number 8, which is environmental influence. Environment has a lot of role to play in determining the genetic makeup of an individual. And we made mention of Siamese cats in cats, whereby the normal four of that is hair, so will be dark. You can have the dark in the tail, you have it in the paw, you have it in the hair. But if the temperature is warmer, you just discover that this darkness will become so inactive production of Siamese cat. And number 9, I made mention of anticipation, which is another condition that the inheritance pattern might not actually uh, obey normal Mendelian law, and that is a case whereby you have CAG repeats of CAG repeats in that individual's genetic makeup condition, that one can predict whether the next 1 or 2 generations they are going to have they are going to suffer from a disease which is called Hutchinson disease. So, and the last one, which I mentioned as a deviation from Mendelian law, is a genetic linkage or genetic mutation, which really happens during meiotic division, especially when there ought to be separation at anaphase, when the anaphase 1 in particular, when all the chromosomes will be pulled apart to the northern and southern pole. So it is very possible that before that time there must have been between 2 chromosomes, and the inheritance of any gene that is the product of genetic linkage or genetic recombination will not necessarily follow the pattern as it was put up by cases whereby Mendel's law might not actually be in operational. So you need to know this, so that when you are given any genetic problems, or questions, so the ratio will actually tell you if it is a deviation from Mendel's law. You will see that it is not going to obey what you know to be 1:1, 3:1, or 9:3 ratio, as you have it in diabrid inheritance, according to how it was put forward by Gregor Mendel. So we have come to the end of today's class. Thank you. Don't forget to